recording. Alright guys, let's go over the ones you guys have any questions on. Any questions, let me know guys. I mean, let's look at the first 10. Questions, let me know. Just give me the numbers on board. Any questions?
9.
9. Alright, what else? Anything else? 10. For 10. Okay, anything else guys, let me know. First 10. Out of 37. 9 and 10. So, I don't know. I saw a couple of you guys got number 2 wrong, and I wasn't really sure why you guys got that one wrong against number 2. So I think that's one of the stuff that we talked about. You guys did okay. I think. Let me see. Number 4. That is the kinetics question. You guys did okay there. And number 6, number 7 guys. Okay, number 7. You guys got number 7 wrong. I mean, some of you guys got it wrong, I think, right? So the lysine with the glycine. See, they're asking what the hell is what's the expectation here? So, so this is where, guys, I don't 100% agree with some of the questions. I mean, I will say that a couple of times, guys, as we go over some questions on Olympiad. Olympiad questions are not 100%. Like, it's not cleanly vetted out. So when it's like college board questions are very, well thought out, and they make all the students take the test, solve the questions before it actually becomes the actual question on the test. Not so much for these, okay? For Olympiad questions, guys, they make it, it's good enough, and it's just like there's typos, that there's a mistake with the answer key. So there are a lot of questions like that, by the way. So number 7, right? So the replacement of the lysine with the glycine in a protein can result. So you guys should really know what the glycine is. Glycine is the most simple amino acid that's out there. Okay, the side chain is just hydrogen there. So the lysine. So the lysine. I mean, I kind of made a little note on the answer, guys. So lysine is a basic amino acid with a positive charge at the side chain. Okay. So it's clearly, I mean, the best answer here. They say except here. So it's what? The loss of negatively charged amino acid side chain. Okay. So it's a positive side chain, not a negative. That's why I thought D is a good answer. But I'm not sure what other answer choice you chose, guys. But it could, I mean, B could be a good answer as well, I believe. Okay. If it's not D, I thought B might be a good answer because, as we talked about, the secondary structure of the protein, which is so. Let me tell you guys. So the secondary structure, what can impact the secondary structure is what? Yeah. So one amino acid that we talked about, it can potentially impact the secondary structure. I think we talked about, which is proline. Okay, that's one of the amino acids that we talked about. This one is less likely to create like an alpha helix or beta sheet. Okay, it's like that's what they call it. So one of the question has it, right? It's one of the helical breaker or something. So it's one of the choice had it. So so I was expecting a question that like question like this, like lysine versus a lysine. I mean, so would that have an impact on the secondary structure? I don't like the question, but unless like something like proline was like brought up, the amino acids, which can have an impact on the secondary structure, but clearly there's a better choice, guys. Like in case, like even though like B, but clearly I think D is a good answer. But that's why I keep on saying, guys, you gotta know your amino acids, okay, for this exam. So a lot of questions you guys got wrong. I mean, it's about the amino acids, so make sure you guys have understanding of what they are and memorize each and single one of them. And you guys, so I think number 8, guys, also as well. I saw what you guys did. Some of you guys got it wrong, I think. Okay, so I think A and C, those are called like purines, as we talked about, right? So which means like it's two like rings structure, as we talked about. So the question is asking you guys, the cytosine will pair with the guanine, as we know. Okay, so they're asking which one it is. I mean, at least either answer you gotta choose A and C. Okay, because I mean again, and I think one of the stuff that we talked about, I think D. I think that will be. I think one of the stuff that we talked about, it has an amine group, which is what? That's the thymine. Okay, and I believe, guys, E is the uracil, and B is the is what? I'm sorry. I mean, D is a cytosine. I'm sorry. D is cytosine. E is uracil, and B is thymine. Okay, so I think that's where it goes, and A should be adenine, and C is a guanine. Okay, so like when you see it, I think you should really know what it is. Okay, so so this is Olympiad. Okay, so AP will never have that question. They will give you what it is. They will even not give the structure for this. But I think the easier way to look at, I think, is as one of the stuff talked about is cytosine. I mean, cytosine, uracil, thymine. These are all what we call pyrimidids, right? They have one ring structure only. And then I think cytosine has that amine group at the top, or like NH2 group, guys. That's one of the ways you can tell. And then the thymine has a CH3 group on them, okay? So one of them. So I think D and B, they're easy way to kind of tell which one it is, okay? So number 8, I don't know how many students got it wrong. I mean, I can go back, but like you gotta memorize. Who knows what the questions will be? But you gotta know your structures, okay? Like uniqueness. They will never, dude, it's a multiple choice. They're not gonna ask you to troll it, but when you see it, you should know what it is. Okay, that's what it comes down to. And number nine, guys, okay? So I don't know. The question is asking pepsin. Pepsin is the enzyme that's in your stomach, by the way, okay? So I honestly think, guys, this is not this is a wrong. I think this is incorrect, by the way. We'll talk about it, guys, when it comes down to the animal physiology and digestion, by the way. So the enzyme that typically breaks down, this is not your first time taking bio, that breaks down protein is what, guys? It's a pepsin in your stomach, okay? And also like trypsin, chymotrypsin, okay? So like peptidase, I mean, those are the enzymes that you see that breaks down protein into amino acids, right? So the first thing first, guys, when these pepsin enzymes are available in their stomach, okay? It reacts or it occurs, it behaves the best or reacts the best in an optimal pH lower than 2 or 3, somewhere out there. So, but pepsin, the enzyme itself, doesn't really break down, okay, like the protein into amino acid. That's not how they do it, okay? So when you guys eat meat, right? So the ultimate goal of digestion is what? Like break down them into amino acids. It doesn't occur with pepsin. Pepsin doesn't do that, okay? Pepsin just kind of soften the meat a little bit, guys. So higher level, they break down more or less a quaternary and tertiary structure at most, okay? And then like going further down, like in the small intestine, where they really break down the secondary structure and even the primary, where the sequence goes. So the question states that, okay, activity of pepsin. Yeah, pepsin really doesn't go break these peptide bonds. So it's not a very good question, by the way. How the questions are being, I mean, I don't want to keep on saying this, guys, but Olympic questions, I mean, whatever. So they're saying, so let's assume, okay, this enzyme breaks down, okay, peptide bonds, right? That's what happens. And question is asking which bond in the does pepsin hydrolyze? Okay, so which means hydrolyze is the opposite in bio in terms of a dehydration synthesis, right? So which one is it? If you look at the diagram, which one would it be, guys? You got to find where the peptide bond is. And I think I mentioned this during that AP bio class, okay? So then if you got to understand, guys, where, so this is basically the end result, okay? That's the end result of the dehydration synthesis, okay, of the amino acid, linking together. So, and I mentioned this, right? What is this, guy? This is what we call, guys, what? Like right here is called what we call the amide. So you gotta look for an amide, right? So that's what you have to look for. So, and that is where the resultant of the dehydration synthesis is, which means what? That is what we consider as peptide bond, okay? And that is what needs to break. Whatever the bond. So you got to find that guy. I think the answer was what? B's the answer, right? Which is number 2. So that's the amide you're looking at. Okay. So the 1, that's not the bond that you're supposed to break. You have to break from the carbonyl to the amine group. Okay. That's why the best answer number 9. I think B is a good answer. Okay. Any questions? It's one of those questions you guys asked. And number 10, guys. So which statement is not true about the effect of the various condition of the activity of the enzyme? Okay, so I don't know. Like I saw some of you guys got it wrong, and I don't know why. Okay, so I saw some of the responses that you chose. So they're saying not true. Keep that in mind. Okay, so I thought A makes sense, right?
I thought it was true.
Yeah. So I thought like these obviously answered, by the way, because enzyme activity generally reduced by the increase of enzyme content. That's not true, okay? If the enzyme, there's more enzyme, of course, the enzyme reaction goes up, okay? So that's completely not true statement, guys, okay? So, All right, that's the first 10, guys, okay? So let's take a look at questions from 11 through 20. Any questions? Guys, give me a second. Let me get some markers, guys, okay? Give me a second, guys.
12.
Come on, yeah. To, whatever, 11 to 20, guys. Any questions there?
20.
20? Anything else?
12.
13.
Anything else? Questions, guys? Huh? You guys should dude, you guys should have the questions ready before you guys come in, okay? So when I ask you for numbers, like right away, okay? Questions later, guys. And by the way, okay, these are what, 2004 to 2013? I tell you guys, the recent questions are much harder. So yeah, okay. So these are so you guys have to do really well on these things, okay? Alright, so number 12. So yeah, the question is asking guys, which of the following is not true of the allosterically regulated enzyme? So this is where I really didn't get to talk about, even for the AP class, guys, so you know. So let me explain to you guys, what this is, allosterically regulated enzyme. So in terms of allosteric, regulated enzymes, right, inhibited enzymes or activated enzymes. So I think the best example you can possibly think is hemoglobin. Okay, hemoglobin is a tertiary structure proteins, right? So which means there is what? There's there are many subunits of these guys, because there's an alpha subunit, there's a beta subunit on these guys. Right? I mean, again, I don't want to draw the I can't even draw them, but so if you look at these guys, okay, so I mean, I think for questions like this, like inhibitions, or even for the activation guys, you might want to have an enzyme as an example, by the way, but I'm just going to give you a real life example in a little bit. Okay, so the example I'm going to show you guys right now is allosteric, okay, cooperativity. Okay, so I think that this will explain the sigmoid, this graph. I don't know what you call that graph, guys. I think it's choice E, guys. When they say sigmoidal shape, how do they have that kind of shape, by the way. So what you have to understand first of all is this, okay? So in terms of when they start to talk about allosteric, okay, it's somehow one of the protein, okay, the tertiary structure protein impacts the other guys, okay. So basically what will happen is, okay, so what do they do, guys, hemoglobin wise? What do they do, guys? They what? They bind to the O2 gases, okay. So basically, guys, if the hemoglobin, there's 4 subunits of these guys, okay, so if one of them, okay, binds to the O2 gases, okay, then what happens then? So basically what happens is, of course, when O2 binds to one of the protein here, one subunit of the hemoglobin, what happens, guys? Yeah, dude, the shape of one of them changes, okay, which impacts what? All the other guys, okay? So, which means that, okay, so what happens then, okay, so then O2 gas, affinity for the O2 gas will increase for these guys, okay? So which means what? They all of a sudden, because of structural change of one of the proteins, one of the subunits, they start to have a greater affinity for the O2 gases, okay? And that is what they call cooperativity, okay? So which means, I mean, if you look at from this, so basically, guys, in terms of the O2 saturation, if you look at it, guys, so this is an O2 level, okay? This is hemoglobin saturation, okay, how much of the O2 gas it has. And now if you look at it, guys, as the amount of the O2 gas increase, what happens, guys? Yeah, dude, okay, so this what will happen is, I mean, one of the O2 will be bind to one of the subunits of the hemoglobin, and it becomes what? It increases, okay, a lot quicker. And this portion represents what we call the cooperativity. Okay, that's represent the sigmoidal graph pattern. So clearly, this is what you're not seeing anything like this. Okay, that is what we talk about the Michaelis-Menten diagram, the graph that we saw, which has an equation that we talked about. So, which means I kind of levels off. This is what we call the Vmax, okay, the speed of the reaction maximum. We talked about that, guys, by the way. So it clearly doesn't follow that for the allosteric process, okay, so okay. So it's kind of far away from that. So and also same thing, guys, the allosteric inhibition as well, like allosteric inhibitions, guys. So if this is an enzyme, if like this is allosteric enzyme, so whatever the enzyme it does, whatever their activity it does, right? So I don't know what it is, guys, by the way. But allosteric enzymes, I mean, there's a reading that I gave you, right? So there's an active form, okay, and there's inactive forms, right? Inactive and active, so it kind of toggles back and forth with the active and inactive, okay? But now, what it comes down to is, okay, so these enzymes rely on some other like molecules for the activations, or even for like cooperativity or inhibition, for example, guys. So basically what happens is, okay, so this is an inactive form, so I'm gonna just give you guys like box shape, okay. So when the enzyme is in this form, it's inactive. When they're in this form, it's active. Okay. So basically, what will happen is, so for example, guys, this catalyzes a reaction. This enzyme, whatever this enzyme is, catalyzes reaction A and B. Okay. A turns into B. So when it's active, okay, there's more of B, for example. Okay. So basically, what will happen is, if there's a lot of B produced. Okay. B is like a particle, right? So B will allosterically inhibit this enzyme. So which means like B will bind to this enzyme somewhere in the enzyme, okay, not on the active site. Okay, its active site is like somewhere here, but the substrate will bind, so A will bind to the active site, right? But now, which is a product of B, right? Which means like B can bind to a site, okay, allosterically bind, okay, which is not the active site on this enzyme. There's a better diagram on the reading that I gave you, right? Which will put this enzyme in what? In a more of an inactive form. Okay? So which means that like B will bind to allosterically to this enzyme, which will keep it in inactive form for time being. Okay? So, and this is not like irreversible. I mean, it's not like irreversible. Yeah, this can pop out later, right? So, but it's gonna toggle back and forth, and also like if there's a lot of A, then A will bind to allosterically to the active enzyme, and will keep it in that way. Okay, so that's what they call, guys, allosteric like activation or allosteric inhibition. Actually, which is they're going to bind to a site other than the active site for these subunits, and then keep it active and more like active form. Yes.
It binds to the inactive, right?
What's up?
It binds to the inactive enzyme.
So it depends, right? Depends what, like for example, different particles can bind to the inactive part of the enzyme, or it could be bind to the active. So if you think about it. This is like more like a homeostasis sort of thing, okay? Which means, think about it, if there's a reaction, A turns into B, right? So in like feedback mechanism, that's what the part of the reading represented, right? So if you have a lot of B, whatever the B is, what the product is, if you have too many products, which is B, what do you want to do? You want to stop production of what? B. So then if you have a lot of B, what happens then? B will bind to the enzyme, okay, allosterically, okay, which means if B binds to the enzyme, what happens? And it will make it inactive. So you can make an active enzyme inactive, okay, or you can keep the inactive enzyme inactive, okay. So that's what they call like allosteric inhibition. Where a particle can bind to the enzyme, like enzyme like this kind of enzyme where it has multiple subunits of the enzyme, and then not permanently but reversibly inactivate the enzyme like this. That's what they call allosteric inhibition. Okay, and it could be activated as well, right? So basically, there's a lot of A, not a lot of B. Okay, what happens then? B will bind allosterically to what? Make the enzyme active and let the the enzyme act so that A turns to B. So my drawing, guys, and there's a better reading on this one on the textbook that I gave you guys, okay? So make sure you guys read the textbook, guys, by the way, the reading I provided for you guys. So the question is asking, guys, the following does not show allosteric regulated enzymes, because I think it's clearly, guys, the best answer was D here, because it followed the Michaelis-Menten kinetics, which is the graph that I presented, guys, over there. It doesn't follow that graph, okay? So I think the best answer, guys, was number 12, because they're saying not true. The best answer is D's answer, okay? All right, so let's take a look at. Give me a second, guys. I gotta charge this laptop. It's a lot of power. All right, let's go to guys number 13. So you guys all did AP Chem last year, so I thought you guys, I don't know. So the question is asking you guys, whatever this thing is, like a 3 domains, which is tightly packed, blah, blah, blah. And the question is asking, if you want to determine the subcellular location of the property folded, whatever protein, fluoride microscopy, which of the following emission wavelength would you monitor to excite 240 nanometers? So like, I mean, if you look at it, guys, it absorbs 240 nanometers. Okay, so one of the things, if you have seen some questions here, if you absorb the light, okay, which will what, excite the electrons, okay, electron comes back down. Typically, if you look at it, what happens, guys? You know, it gives you what? A little bit greater wavelength, right? So, you know, it kind of gives you, I mean, it loses some energy per se, I guess. I mean, it doesn't really, you know, conserve the energy too, you know, very well, but it's so, domain A portion of the protein, okay? It can absorb somewhere around 230 to 270 and gives off what, 280, 310, okay? So they're saying, it's very clean cut question, right? They give 240 nanometers, okay, which is what? In domain A absorption, right? And then it emits, okay, around 280 to 310. So it should be that around that range, I think, which is what the best answer is around. So you can say, what about B then? Okay, so I think A is right in the middle of it. That's why A might be the best answer. So, you know, so that's why it's a good answer. I mean, it's one of the terrible questions, guys, you might be getting. So, you know, so
between A or B.
yeah, exactly. So I think it's not only you, man. I tell you, it's just, you know. So, but I think A is being smack in the middle is a better answer than I think B compared to B. So yeah, there's an old expansion in this one. Let's take a look at number 20. So just looking at some of your work. pH was not a problem. Let's see. You guys did pretty okay here. So number 19, I'm sorry, that's a positive cooperativity. That's basically allosteric, interaction between the subunits. So it's like how we talked about for the O2 gases, you know, hemoglobin. Number 20, guys. So number 20, guys. So it's one good thing to learn, guys. I mean, so it's all of them is true, okay? So which means like you can look at the choices, guys. I mean, A, B, C, D, and E, they're all true, because we know. So yeah, I mean, I don't know what else to say, guys, on this one, just so you know. So it's for A, okay, tryptophan, tyrosine, and phenylalanine, okay? When they say, okay, aromatic amino acid, so I don't want to really, guys, get into chemistry, guys, what aromaticism is, okay, just so you know. So if I remember this correctly, I forget it also too, guys, by the way. But now, what's the best example of, what's one of the things that you know that's aromatic? Like this is like SAT bio stuff.
Benzene?
Exactly, yeah, right there, benzene, right? So benzene is what, guys? Benzene looks like this, right? So what do you guys know about the benzene, guys, by the way? So benzene has what? Every other bond is double bonded, okay? So, you know, that's what we got. So then let me ask you a question then, okay? So Derek, let me ask you a question then. Is this aromatic too? Is this aromatic?
I'm not sure, but I'm going to guess now.
What do you think? I mean, like, what, you know? What do you think? What makes it? What's the definition of aromaticism? So, yeah, what are these bonds, by the way? What are these bonds? What are these bonds, guys? Yeah, these are what? Pi bonds, right? Right? So pi bond, double bond, right? So single bond is 1 sigma. Double bond is what? 1 sigma, 1 pi. Triple is what? Triple is 1 sigma, 2 pi. Okay. So if you look at it, guys, they're pi bonds, right? So what defines, okay, aromaticism is what? You got 4n plus 2, okay, pi electrons. Okay, so that's what defines, okay, aromaticism. Okay, so this molecule is aromatic, the first ring structure, by the way, okay, and then 4n plus 2. Okay, so is this aromatic? Yeah, because what? You got 1, 2, 3, 4, 5, 6 electrons, pi electrons, right? Do you know what I'm saying, guys? Here, so which means okay, 6 it fits into this equation. So n must be an integer value, because we know n must be an integer value, right? So yeah, this is aromatic. Will this be? So okay, 4 is equal to 4n plus 2. Is there aromatic, guys? No, you can't. N cannot be an integer value. It's not. So that's one of the things you have to know, okay, to decide what the hell is aromatic, what it's not, okay? So This is hard. I understand 100%, guys. Okay, so you gotta know your shapes. You gotta go and know your shape. Okay, so first thing, tryptophan and tyrosine and phenylalanine. Okay, so you gotta go and you gotta memorize the structures, by the way, okay, and see. Okay, so does it follow this equation? Okay, to be determined as aromaticism as a molecule itself. Okay, because some of the amino acids is rings, right? So you gotta go back and see. Okay, so do they have this number of the like pi bonds, right? So which I assume they do because the answer is all A, B, C, D, and E, right? And then like you gotta go back and see which of these A, like B and D, like you gotta see which one it is, I believe. Okay, so it's good. It's one good exercise for this question, guys, because you get to learn some of these amino acids per se, guys. So and I think proline, as we talked about, like helix breaker that we talked about, guys, by the way, because they can't make that regulatory hydrogen bonding for the alpha helix and beta sheet. That's why they're known to be a helix breaker, and they can't really create also the beta sheet as well. Okay. So it's a good exercise, guys. Number 20 in terms of amino acid, like their properties per se. Okay. So I noticed, guys, again, so a lot of questions you guys got wrong is based on the specific amino acids question per se, because you might not know the structures for them to begin with. Okay. So, but there's some question where I saw that they gave you the structure for all, and you guys still got it wrong. So I think that one was number 30. Okay. So some of you guys got it right, some of you guys got it wrong. Okay. So like, do you see that, guys? Like number 30, guys, phenylalanine. Do you see that, guys? How many pi bonds they have? They got 6. Do you see that, guys? Is there aromatic? Yes, they are. Okay. So do you see that, guys? So the question is asking, you guys heard the question, right? The tiny hair on the geckos, like blah blah blah. The question is asking, van der Waals interaction, which is like LDF that you guys know, guys. Okay. At the tiny interference where the geckos which I mean, so they want to have a strong like LDF. Okay. So you want to have the amino acid that's what nonpolar. Okay, so just like so you want to have all nonpolar. So the question is asking, which of the following is what? Which of the following is a polar? Which of the following is a polar amino acid? And clearly there's one answer. That's what? That's the polar amino acid, which is what? Choice E. Okay. So choice E has what, guys? That hydroxyl group of OH, which is what that makes them what? Very polar. They can make hydrogen bonding. Okay. So that's what reason why, guys. Number 30, the E is the best answer out of all. I thought it was an easy question. Okay. And phenylalanine, right? That benzene-looking group, guys. Okay. That is what, guys? So like that is also what? Hydrophobic as well. Okay. So any hydrocarbon, okay, which means like if you got a carbon hydrogen only, guys, they're hydrophobic. Okay. And they relatively make very strong LDF, by the way, okay, because they have a lot of electrons. So LDF, you guys coming from AP Bio, right? AP Chem, sorry. So what's the strength of an LDF? What makes a very strong like LDF, guys? What indicates? Yeah, Indy, yes. What is indication for the strong LDF?
Like big electron cloud.
Yeah, the size of electron cloud, right? Like how big what the molecule is and how many electrons they got, right? So look at this, they got a lot of electrons there, which means what? They should have a strong van der Waals / what LDF there, okay? All right, any question, guys, from 21 to like what the whole thing, guys? Let me know. Any questions for the whole thing? Let me know.
22.
Okay. Anything else?
32.
Any questions?
28.
What else?
36 and 37.
35.
Anything else? So let me tell you guys something, okay? So if anyone of you, when you like your friend or whoever, guys, giving advice on this exam and they took it like way back, even like 5 years ago, questions are completely different. It's much harder now. So so that's why I'm giving these questions as like a practice for you guys right now, because like it's nothing like this anymore, guys. So like the questions you guys get solving is like from 2014, 15, 16, 17, 18. Okay? So those are the ones, and also including the national stuff. So, and these questions are just like, like you have to know these questions a little bit, how to solve them. Number 22, guys, see what it is. And like some of the questions, guys, I clearly think the answer key is wrong, but I figure like whatever, that's the answer key they gave it, so I just left it as is. Henderson has number 22. Okay. This is something you have to solve, guys. This is a if you can question, by the way, a little bit, okay? So you guys, we talked about it a little bit, guys. I mean, they actually give an equation for this question, by the way. It's useful to determine the amount of weak acid and weak base to weigh out the buffer system. It's also useful to determine the percent of the functional groups in its ionized state. If a carboxylic acid has a pKa of 3.9, what percent of the functional group will carry out the negative charge at at pH of 4.9? So I think this is one of the question, guys. It's a chem question, guys. So I don't know if you guys want to go over, guys. I mean, we can go over, but let's kind of look at the other ones first, okay? And then maybe come back to it, guys. Okay? Let's go number 28. Let's go number 28, guys. See what it is. So this is one of the inhibition question, right? So the question is asking, guys, so in the example above, assume that D is an allosteric, like what this is what we talked about, guys, inhibitor of the enzyme E2. Okay, D would, like what the hell would do? So as we talked about, guys, D would bind to an allosteric side of E2, so to make it inactive, right? So whatever that kind of, whatever I said, guys. So the answer was D, guys. The binding of the E2 at the side different from the active side, that's what we talked about, okay? But change the shape of the active site so that B will no longer bind. Okay, that's one of the stuff we talked about, guys. I think it's pretty straightforward what the allosteric inhibition represents. So allosteric inhibition is not very similar. It's very similar, guys, to non-competitive inhibition, but they're a little bit different in terms of how they're, how they act. Okay, so keep in mind, guys, make sure you know the difference between non-competitive inhibition versus allosteric inhibitions. Okay, so keep that in mind, guys. So the similar thing between the two is what, guys? If they bind to the site other than the active site, that's one thing that's similar about it, okay? But when you get an allosteric inhibition, what happens, guys? Yeah, dude, the substrate will not be able to bind to the enzyme, while non-competitive inhibition, they can still bind to the active site, and what? The process might be a little bit slower, okay? So that's a little bit different, guys, by the way. They're not the same thing, okay? Non-competitive inhibition and allosteric inhibition, they're not the same thing. It sounds very similar to each other, okay? All right, let's go to, guys, number 32. Yeah, 32, I mean, I don't know. So some of you guys got it right. Some of you guys, like, perhaps did not. And then I should have put this question, guys, in this chapter, in the chapter I'm going to go over today. So the question is asking you guys, which of the following is a plausible structure of the lipid to make up the lipid monolayer? So keep in mind, guys, here, so cell membrane, right? This is the outside of the membrane. This is outside. This is inside. Okay. So out and in, right? So if you look at it, guys, I mean, they always need to have what? Outside is water, as you guys all know. Water is outside, and inside is also water as well. So, what's something significant, guys, about the cell membrane? They have what? They have a zone of hydrophobic zone, right? So basically, how they do is what, guys? I mean, they have a bilayer. A cells have bilayers, which means that they have what? A phospholipid, layer at the top. Okay, phosphate, which is the polar side facing this way. Okay, non-polar tails facing what? The membrane side, right? Like the inner side, right? So that's basically how it looks. But they're saying, okay, so they can't have a bilayer like this. They're gonna have a monolayer. Okay, so it doesn't matter what it is. They still have what? They need to have two sides that are polar and what? A non-polar side here. So you gotta find a structure, okay, where what? You find a polar side on both sides. One side must be polar, and two sides must be polar, and the middle portion must be what, guys? Non-polar. So hydrophobic, right? So that's a structure you have to kind of find which one that kind of fits into that category. Must be the answer, guys. So A is so A is what, guys? What's A? A가 뭐야? What's A, guys? A is what? That's a phospholipid, okay? Except for it's missing the choline sector of it with nitrogen there, but that's a structure that you guys know probably of what, guys? Like phospholipid, right? So no, no, I don't think so. Well, what is wrong with B, guys? What's wrong with B, guys? I think B is If you look at D guys, I mean, so yeah, they got the zone of the hydrophobic, yes, they do. But what happens, guys? There's no hydrophilic side. There's no like, you know, like there's no OH group. There's no like positive or negative side that can be hydrophilic. So it's really out the door, guys. And part C as well, guys. I mean, they have a long chain. I get that, okay? They have long tail. I get it, guys. But still, this one is what? This is a missing. On one side, they have a phosphate group, but the other side is what? Still just long tail of hydrocarbon, which is not the hydrophilic side. So you got to look for the one that's what? Both sides of the end of the molecule must be polar and polar. So I don't think C is a good answer. I think D might be it, right? D might be it because why? On one end, they got what? OH groups on them, right? On one end, they got what, guys? OH group on them. And on the other end, they got what, guys? They got a phosphate group on them, right? So on both end of these molecules, what? They got what? A polar side. In the middle, what? On the middle, what do they got, guys? They got what? A long chain of hydrocarbons, which is hydrophobic. So that's a perfect one you can possibly choose as well, guys. Yeah, this can be a monolayer, right? All right, let's go to guys number 35. 35 guys. This is recording, so I can't say these things. All right, so 35 guys. Question is asking, which of the following guys, of the following amino acid, which is least likely to form a strong interaction with the other amino acid? So, like they're saying, whatever this amino acid is, it failed to produce like you know, LDF or like ionic or like, you know, like hydrophilic interactions, like, you know, like they can't do anything like that. Okay, the question is actually which of the following is like that. So I thought the answer is what? Like very clean cut is what? Glycine. Because why? Glycine is what? Glycine, if I'm not mistaken, guys, I don't memorize amino acids by the way, okay? They got this only. You see that, guys? Yeah, the side chain is hydrogen. Yeah, what can they make? They can't make like LDF. They can't really do much with this. Okay, they just got a single hydrogen there. Okay, so which means what? Yeah, dude. Like, okay, so what really can they do? I mean, size is small. I get that. Okay, but that's about it, right? So that's why I thought, guys, the best answer is what? D's answer in this one. The question there. All right, let's go to guys number 36. I mean, dude, I wasn't very happy for number 36, by the way. So I saw a lot of you guys got number 36 wrong, and like I was like, what the hell? So that's something that I understand, guys. Like maybe I wasn't clear enough in this question. So number 36 is asking, guys, what's the following aligo? What's aligo? Yeah, when they say aligo, what does it mean? Yeah, so I don't know if you guys are good at English, guys, by the way. Much better than me, probably. When they say aligo, like lipid, aligo, like whatever. Yeah, what does it mean, guys? What are you guys saying? Yeah, what do you think?
Not sure.
Yeah, what do you think?
8.
What?
8.
Huh?
The number 8.
Aligo? I don't know. What do you think? It means what? It means short. Short chain. Okay. It's not long. It's very short. So you Asian, no? Yeah. All right. So I mean, oligonucleotide means what? I mean, they don't apply this term for DNAs only, guys. This can be applied to carbohydrates and proteins as well, guys. By the way, okay. So highest melting point. So I don't like the term. So just like a melting point, I don't like it. Okay. So you guys should be smart enough, man, to figure this out. I thought you guys were the top kids in Jericho or something. Okay. So like the term, yeah. When they say melting. Okay, like dude, like I mean, how the hell do you melt? I mean, this is not a solid, right? So yeah, like DNA, right? When they say melt, yeah, what does it mean? Breaking of these two guys. Okay, breaking the two, portion of the amino acid, right? So the question is asking, guys, in the pair with the proper complementary strength. So they're showing you only one of them, right? So if they're paired with like proper, so meaning, guys, if it's A, like it's paired with T. If it's C, paired with G, like like that's a proper pairing, right? And then what did I say, guys, in this one? A, I definitely mentioned this, okay? Creates what? 2 hydrogen bonds with T, right? What about C? Makes what? 3 hydrogen bonds with G, okay? If you want it, right? So what did I say? If there's a greater content of C and G versus A and T, what happens, guys? Yeah, dude, this is all harder to break. Because what? More energy must be put in for this, right? So in terms of separation of the two strings, if there's more C and G, what did I say, guys? Yeah, this is harder to break, right? And I think that's what the question is going, right? That's why the answer is what? C is the answer, because why? It only has one C and G only. Okay, this is these are freebies. Okay, these are like you know, like take it, go ahead, you know. That's number 37, guys. Yeah, this is a question. So if you look at it, like I personally do not agree with this question. Okay, the answer from this question. Okay. Below are the structures of the five amino acids. So I on the answer, like you know, response, I gave you guys like what each of them represents. Okay, phenylalanine, glutamic acid, like blah 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 blah, right? And the question is asking, if you look at it, right? Yeah, let me ask question, guys. Is histidine on choice C, guys? Is that aromatic or not? What do you think? Is it aromatic or not? What do you think? It's not, right? Because why? It doesn't fit into that 4n+2 ratio, right? Question is asking guys like, what the hell do you know about it? Okay, kind of significant stability of the tertiary structure of the protein. So maybe like you know, I don't know. So tertiary structure is what, guys? One of the stuff that we talked about is side chains, right? It's dealing with the side chains. So so the hydrophobic side chains will create what? Hydrophobic interactions, you know, with each other, right? Like LDF that we talked about, okay? Hydrogen bonding is one of them, ionic is one of them, right? So you know. So, but one thing the bio keeps on saying is what, guys? Like hydrogen bonds, right? Hydrogen bond is really weak. Okay, hydrogen bond is like super weak, right? It's not a strong bond, according to them, right? Ionic for them is also weak bond. Okay, like I mean, dude, like in bio, they consider ionic bond as what? Relatively weak bond. Okay, ionic interaction, weak bond. So do I agree? Absolutely not, but whatever. They think covalent bond is very strong. Okay, in bio, covalent bond, guys. Okay, relatively strong bond. One of the question I hate to hear every time is what? Oh yeah, in bio, like dude, like they ask you, which one's stronger bond? Is it ionic or covalent? Which is stronger bond? I mean, that is not a good question, okay? That if we talked about this in the chem classes, guys, they cannot ask the questions, okay? That's a stupid question to ask, okay? But they still do, okay? No. But now, this is strong bond, according to bio, okay? And now, one thing I made sure is what? There is tertiary structure. There is an amino acid that can make this, okay? Which one was it? Which one was it? Which amino acid can make this? You know, it's a tertiary structure, okay? What did I say, guys? You gotta remember these things, guys. Cysteine. Cysteine is one of the rare amino acids that can what? Create disulfide bridge. Okay? Which is what? Covalent bond. Okay? So this stabilizes the tertiary structure of the protein. Okay? Which what they say in bio textbooks and all, stronger than these two. Okay? So then so with that, is that the answer? I mean,
I put cysteine.
Yeah, cysteine. That is my dude. That'll be my answer, but it's not the answer. The answer is A. Okay, so answer. So
That makes sense.
yeah, exactly. Like I would put A as the answer. I mean, I would put cysteine as the answer. D's answer. Okay, because what? Look at the bond, right? But now they say what? They said I think A, which is phenylalanine, as one of the stronger. Because why? I mean, same thing, right? Because they have that shape, right? Because what? This can make what? Stronger LDF. Okay, so for whatever reason, they think this is stronger than that guy. Well, do I agree with this? Absolutely not. Okay, so I rarely say this, by the way. Like with the test makers, I don't try to argue with their answers. But again, I don't know. Like yeah, this is goes against what I think the answer is. Okay, I think one of the questions is the same thing. Like so, according to you gotta see the patterns here. Okay. So for them, when they see stuff like this, right, like when there is like a benzene group, or when they see something that's aromatic, they think, okay, so you can have a strong LDF, which can contribute to a lot of strength in terms of structure perspective. So that's one of the trend, guys, for the Olympiad question, which I do not agree with. Okay, so that's why they're putting it as the answer, guys. Yeah. So there was a similar question like that you guys didn't ask. Like all of you guys got it wrong, I think, or most of you guys got it wrong. There was something about. I don't know. There's a question that the stability of the soluble globular protein or something. I don't know that you guys got wrong. I don't know which one. So that's one of the examples, guys. Okay. Let's go back to guys 20 something or whatever.
21.
21. 21. Yeah. 21? Yeah, right there. So 21, guys, structure stability, like, I mean, hydrophobic effect, I mean, it's a little bit different, by the way. But, yeah, so it's a little different question, because I think this question is a little valid because they said this is a soluble globular protein, which means like this will, like, you know, dissolve in water, but what keeps them the integrity, the shape of the protein, because what? You have the strength of the hydrophobic molecule, hydrophobic interaction inside, you know, that portion that's non, like, you know, soluble. So this question made a little sense, but there's another question that there's some other question that I've seen. I don't know which one. All right, so let's take a look at, guys, number 43, which absolutely, you know, a disgrace for some of the folks who got it wrong. Who asked this question, by the way?
I did.
So, what? So I'm just gonna set it up because I don't have a calculator here, okay? So I'm just gonna set it up for you here. So the question is asking, right? What on English? Question is asking, pKa of 3.9 for the amino acid, okay? Well, carboxylic acid for the amino acid, whatever, okay? So the question you guys should know is what? pH is equal to pKa, okay? Plus log of base 10 of concentration of A minus over HA, okay? Conjugate base and the acid, okay? And what they're saying, pKa is 3.9, okay? pH is 4.9 or something, okay? And log of base 10 of like the ratio or something, okay? So, so if you look at it here, it's buffer zone, right? That's what you got. So so they're at half, remember this? Half equivalence point. That's when what? Concentration of their equal. Okay, right? This is when like pH to pKa value, right? This is like, but we are beyond that point, which is like we're here. Okay, which means what? Dude, like there's what? There's greater amount of what? A minus over HA. Okay, there's more of this guy at this point. Okay, there's more of the minus charge ion, let's say, okay, conjugate base compared to the acid. Okay, by how much? Yeah, like dude, like you can what? Like I mean, you can solve it. I think. Okay, this is subtract one here. One is equal to a log of base 10 of the A minus over HA. Okay, I think you put 10 here, right? 10, like get rid of this log, right? So 10 is equal to the log goes away, A minus over HA. Okay, so if you do the concentration of A minus over HA, that's what then you get 10. Okay, so there's a lot, there's like 10 times more of this guy, okay, compared to that guy, okay, compared to this guy, I'm sorry. That's what it is, okay? So you pick the appropriate answer for that one, whichever that is. All right, so any other questions? Let me guys. So like you guys have to be like, I don't know, like perfect with these questions. So like, that's my concern, okay? If I go to your result, okay, so I'm not asking something to be like, I'm not asking you guys to get like everything right, but on average, you guys got 70% of the questions right, okay? That's on average for you guys. That's not gonna cut it, okay? So you know, like meaning guys, the questions will be harder for you guys when you guys take it. So this you gotta do like, dude, you gotta get 90% of the questions right, okay? And yeah, I mean, you guys have to do better than this one. 70%. I mean, and you guys got it done and all that stuff, guys. By the way, this is a competition test, guys. Okay, whoever that's doing better than you guys, they're gonna make it. So keep that in mind, guys. You gotta perform. Like you guys have to spend a lot more time. Okay, I think based on what I saw, guys, you guys have to put more time, guys, reading and solving questions and all that stuff. Guys. Maybe you guys did, but I don't know. All right, guys, let's take a look at what we discussed, guys, for the next chapter. Next chapter, guys, we have to go right away. I lost 50 minutes for this. So, guys, the chapters that I'll send you an email about it, guys, as well. But the reading, guys, that you guys have to do on the next chapter, guys. I'm using my book, guys, like at home, but that's as a different chapter. Chapter 6 and 7 is for you guys, okay? For next week. Chapter 6 and 7, guys. Homework wise, that's what you guys will do. That's the tour of the cell and the membrane structure and functions. Okay. And then next week, we'll do respirations and photosynthesis. The week after, we're gonna do a cell communication, cell cycle. Okay. So, and then after that, we'll go into genetics. Okay. So that genetics is tough. I tell you, it's tough on this exam. But I like genetics because they're very solvable, actually. So on the test. I mean, if you know what you're doing, you can solve them. Okay. So instead of like memorizing like the amino acids, by the way, like instead of memorizing, oh yeah, like phenylalanine, what's the shape, guys? But this is like you solve them. You apply what you know and solving rather than memorizing it. So I tell you, man, like you gotta know your amino acids, guys, for this test. All right, let's take a look at it, guys. The next stuff we're discussing as we go. So today, guys, the first part. I mean, I have to kind of. I made a lot of notes for today, guys. By the way, I think I gotta like shorten this out a little bit, okay, to finish this on time. So first thing first, guys, let's talk about the cell organelles, okay? So I'm not going to explain to you guys like the cell, the organs, stuff like that, guys. That's on you guys, okay? So you guys probably learned that in like regular bio, so I'm not going to do that, guys. So maybe I'll reiterate a little bit, guys, in the AP class, by the way. So that's when I have a little more time to talk about some of the easy stuff, right? So cell organelles and like the functions and like what the hell they have and the details and so organelles, and we're going to discuss, guys, the cytoskeletons, and we're going to talk about the cell junctions and cell membrane overall and transport today, okay? So I can't really go into much detail because we don't have time. But in terms of organelles, so first thing first, guys, we have prokaryotic cell, okay? Prokaryotes, okay? We have eukaryotes, okay? Make sure you do the reading, okay? So just let you know. Dude, like I tell you, man, like I read it twice, okay, before class. Like, dude, like the whole thing, the like 6 and 7, guys, the whole units, whatever you call it, guys. Like I read it twice. Like I read it on Sunday. I read it like this morning, guys. So it's not that I have like all day, guys. Okay, but like, dude, like you gotta read the stuff, guys. Okay. So prokaryotes or eukaryotes, guys. By the way, you gotta know the difference here. So prokaryotes, guys, they don't have nucleus. By the way, I mean it's obvious, right? There's no nucleus. Eukaryotes, guys, they have a nucleus. Obviously, no question about that one, right? So, prokaryotes, guys, I mean, they got one organelle, which is ribosome. Ribosome is the organelle that they have. That's in common, okay? So, so just later, guys, I mean, they got the ribosome. That's all they got. Their DNA is all over the place, guys, as a nucleoli, by the way. They have a plasmid. That's a question for a different day. So, and eukaryotes have ribosome also as well. Okay, this is something that's common between those two. Okay. And keep in mind, guys, by the way, and college board make this mistake also, guys. Not all prokaryotic cells, okay, like are like the same. Like meaning, guys, in the classification chapter, guys, there's something called domain, okay. In domain, guys, okay, there's a 3-sector of domains, okay, which is eukarya, which represents eukaryotic cell, by the way. There's archaea. You have to write this down, okay? There's like a bacteria, okay? So they are not the same thing, okay? Archaea and bacteria, guys, they're not the same thing, by the way. So a lot of people think, guys, okay, when they say prokaryotes, they only look at this one, bacteria, okay? That's not true, okay? So they're very different, okay? Not very, but there's a difference between these two, guys, by the way. So we don't talk about it right now, guys, by the way. But but a lot of times, guys, even college, where they make a mistake that prokaryotes only refers to bacteria only. Okay. But question for the day. But the ribosome, do they have the same ribosome, guys? Do you think their ribosomes are the same, guys? What do you think? They're not the same. Okay, their ribosomes are different. So the prokaryotic cells, okay, the ribosome is what, guys? They call this a 70S ribosome. Okay, this is an 80S ribosome, by the way. Okay. So what the hell does it mean by 70S and 80S, guys, by the way? Okay, this is called a sedimentary unit, like a sedimentary unit. So the S called, so it's called a Svenberg unit. Not that you have to know them, just so you know. Not even for the Olympiad, by the way. So basically, what happens is, guys, here. So it's a smaller, then that's larger. Okay. So the ribosomes have a smaller unit and smaller subunit, larger subunit. Okay. Same thing. They have a smaller one. There's a larger one, by the way. Okay. So these subunits, guys, and they're made up of, I think, 50S subunits, 30S subunits. Okay. 50S and 30S. This is a 60S. This is 40S. Okay, so this is they're slightly larger than these guys. Okay, by the way, I mean, you guys know what ribosomes do, right? They're the site of what? They're the site of protein synthesis. Okay, that's a whole sector of it. Okay, a whole section is on that one only. Okay, protein synthesis. So we have to go into details today. So, what makes up ribosome? Okay, what makes a ribosome? That's what it's in there. What the hell makes it? It's what? Ribosome is made up of rRNA plus a bunch of proteins. Okay. So ribosome is made up of the RNA and protein only, okay? So keep in mind, guys, you will see, okay, as the chapters go by, okay, that the RNAs, a lot of times on the RNA acts like a catalyst, okay, like it acts like an enzyme, okay? Ribosome. Even ribosome, right? RNA will decide. So here, like it does a lot of enzymatic activities. Okay, so what do we call? Okay, you guys have to know, what do we call an RNA act as an enzyme? It's called ribozyme. Ribosome. So like this is kind of goes back to the conversation that we had. Okay, so back in time, man, like when things like were like in cells or even the living things were very, very simple, okay? They're not even able to create a protein for most of the cases. They used what? RNA. For what? As an enzyme, okay? That's why they can act as a ribosome, because we know. So why? Okay, they have the same thing. These guys, same thing. Okay, they have the RNA, so they might have a little different ribosome RNA, but the proteins, by the way, even though the ratio between these two, but what? The same thing. Okay, so in terms of evolution perspective, okay, the structure of the ribosome and what's in the ribosome and their function is what? Very conserved. Okay, which means what? Yeah, dude, like they don't mess it up. Okay, so evolution tends to change a lot of things, right? Evolution, guys, I mean, so whatever that makes them better, evolve better, and make them more competitive, guys. But ribosome didn't change a lot from bacteria to human, which means what? Very conserved, which means what? Yeah, like there's not much to change. So keep in mind, guys, here, okay, so this is 50, by the way, and 30. So they don't, they don't add up to this, okay, so yeah, if you guys wonder, okay, so this is like, oh yeah, is it 100? No, so you know. And they have a smaller subunit, there's a larger subunit, guys, by the way, so ribosome is. We don't have to go very deep, guys, because there's a whole chapter on this one, guys. I'm not going to waste my time on this right now. On the ribosome. So, the other things, guys, that you have to understand, guys, I mean, a lot of the discussion today will be eukaryotes, by the way. The eukaryotes, guys, they got nucleus, by the way, okay? So inside the nucleus, in the cell, okay, this is nucleus, right? So keep in mind, guys, here, this is a nucleus. Okay, this is a plasma membrane. Okay, plasma membrane. So keep in mind, guys, here, entire space from here, so from the nuclear membrane to the plasma membrane, guys, the entire place is called the cytoplasm. Cytoplasm is what they call the region from the nucleus to the cell membrane, guys. The entire region is called cytoplasm. Okay. So anything, okay, found between the cell, like nucleus to here, is called cytoplasm. And the fluid, right, the water stuff, right, that's in there called cytosol. Cytosol is a liquid portion. Okay. So now, nucleus, guys, by the way, it has a double membrane. That's really important. And also, guys, have nuclear pore. Okay. There's a little holes in there, guys, by the way, so there's stuff to go out. And then inside the nucleus, okay, which we assume, guys, our DNA is in there. So eukaryotic cells with their DNA, okay, is found in a form of what we call chromatins. Okay, chromatin. So chromatin is where DNA, okay, DNA, guys, of course, you guys understand, guys, is a very like sacred. They have to keep it, you know, like non-mutated, guys. By the way, they wrap the DNA in histones. It's a protein called histone. Okay, you gotta know this, guys. Histones, DNA is wrapped around the histone, and it turns into what? They wrap it around. There's a whole section. Their whole section, guys. It's found as a chromatin. Okay. Typically in a cell, okay, unless a cell starts to divide, they're found as what? Chromatins. Okay. Keep in mind, guys. Prokaryotic cells, they got no histones. Okay, so you know they got no histones here. Okay. But yeah, this is the problem. Okay. Bacteria, they don't have it, okay? Archaea, they have histones. So that's where, guys, they can't just say every prokaryotic cells don't have histones. They can't say that, okay? Because archaea, they might have histones. So, like chromatins, right? So a lot more to go on this one, guys, by the way. And then the cell is getting ready to divide. They convert the chromatin into what? Something that you guys know, which is a chromosome, right? Which is like, we got like 46 of them, by the way, okay? So this is when the cell division occurs. Okay, they condense the chromatin into chromosomes, by the way, okay? That's what's found in the nucleus, by the way, okay? And then right outside the nucleus, take a look at it. So basically, guys, the stuff to talk about now is basically what they call endomembrane system. Okay. Nothing fancy, guys. Like endomembrane system, guys. It's not fancy. It sounds fancy. So what's part of this endomembrane system, guys? You guys probably heard, guys. Endoplasmic reticulum. Okay. Golgi apparatus. Okay. Lysosome. I mean, also, I could say plasma membrane. So these are part of, guys, what they call endomembrane system, okay? Endoplasmic reticulum, Golgi apparatus, lysosome, plasma membrane, okay? So, a lot of times, guys, this is, even, they asked this on the AP, by the way, okay? Like for example, guys, protein synthesis, okay? So, I mean, again, you guys all know these things, guys, by the way, if it's not first time you're taking bio, guys, by the way. So you probably know, guys, okay, there's a, endoplasmic reticulum, guys, they have what? There's smooth and rough, okay? They have SER, they have RER, you know, smooth and rough. So, in terms of understanding what it is, guys, okay? So, when they have, when there is a protein that needs to be produced, okay? What happens? Look at it. The nucleus send what? They send out the ribosomal RNA, okay, to what? To nucleus sends mRNA. Messenger RNA to what? Ribosomes, right? So ribosome, there are two distinct locations for the ribosome, okay? Ribosome can be found on the ER, which is endoplasmic reticulum, or what? The ribosome can be found on cytoplasm, okay? Like freely floating around cytoplasm, right? So keep in mind, guys, here, so ribosome can be cytoplasmic. There can be, the ribosome can be found on what? Found on the rough ER. What's the difference? The difference is, what's the difference between this guy and that guy, okay? Structure is exactly the same. They're what? They're both the same thing. They're both what? ADS ribosomes. Okay, they're exactly the same thing. Yeah, so what's the difference, guys? The difference is what, guys? After protein is produced or translated, what will they do? Okay, where will it be used? Okay, so if the ribosome, if the protein is synthesized in the cytoplasmic ribosome, which is like floating around, okay, typically this is what, guys? This is where whatever the product is will be used in the cytoplasm. Okay, so for example, what are the reactions you guys know that happens in a cell in the cytoplasm, such as glycolysis, which we'll discuss next week. Okay, glycolysis is something that occurs in the cytoplasm, right? So typically what, like, okay, so whatever what's produced here is what enzyme that what catalyzes a step of glycolysis, which is, God, you gotta know every step of this, by the way. You gotta know every step of the glycolysis. You will never do that, guys, in your school, by the way. So you gotta know it, glycolysis. So glycolysis is what, guys? Turn glucose into what? Into pyruvic acid. Okay? It's a 10-step process. 10-step. You gotta know every step. You gotta know every single step of the 10-step of the glycolysis process, by the way. Okay? So, yeah, dude, like whatever that was produced here is what? Yeah, it could be what? It could be the enzyme in one of these steps. Do you know what I'm saying, guys? Okay? So, guess what? Yeah, the cytoplasmic ribosome is where what? Yeah, whatever the product is, we use in cytoplasm. Okay? It doesn't go out. But now, if the protein was synthesized on the ribosome on the rough ER, okay? Their destination is what typically what? Either the cell membrane or what? As what? Or it could be what? A secretory vesicle, which means what? Outside the cell. Okay, so it goes out. So the protein or whatever is meant to what? Go out of the cell. Why does it go out? It could be what? It could be a hormone, insulin, for example. It goes out. Okay, so keep in mind, guys, here, so if the protein was synthesized on the rough ER, what happens, guys? They typically what? The target is what? Outside, okay? So, if a good question you guys can ask is what, guys? Last week, what did we talk about? Protein folding, right? Which not that, like, okay, whatever. Yeah, protein folding, right? 그게 뭐였어? Chaperone protein, right? Chaperone, right? They require chaperones, by the way, okay? Because what? They're outside. They don't, okay? They don't need it. Because it's inside the ER. Okay? Keep in mind, guys, here, by the way. So so rough ER, it's like a little what? Like little sacs that what? Yeah, like it's a little channel within the cell right outside the nucleus. But it's a little channel to transport the protein that was produced. Okay, that's typically what they do, right? Transportation per se. So now, so basically, guys, this is understanding of what endomembrane system is. So how do they? So whatever that was produced in here, so there, the protein was produced now here. Okay, what do they do with it? So it's meant to go out. Okay, as a secretory vesicle, right? Like to go out. How do they do it, guys? How the hell they go out? So this is the what they call, guys, Golgi apparatus. Okay, like this is like again, like my drawing, guys, is not the best. Look at the textbook, guys, by the way, for the drawings. So this is Golgi apparatus, guys. I mean, they meant to what? They meant to package the product, okay, and modify the product. I'm sure you heard that, right? And then what? Ship it out, okay? Take it out to the cell. So first thing first, you have to ask yourself, guys, okay? From the rough ER to Golgi apparatus, how the hell did they get there? How do they ship it out to them? Okay, so they have the product. How do they get it to them? Through what? Through vesicles. Small, tiny vesicles. Okay, 잘 들어. Well, listen very carefully. This is not the reading, guys. Okay. What they create. So basically, what happens is let me show you guys. Okay. This is from the rough ER. That's the protein they produced. Okay. So basically, what happens is what, guys? A portion of the membrane. So like, dude, everything is like a membrane. Okay. It's like a cell membrane. Okay. So these are they call this, guys, ER Golgi apparatus lysosome, right? Peroxisome, like mitochondria, like chloroplast. Those are what? Membrane-bound organelles, okay? They have a phospholipid membrane, right? So this is membrane for the ER. They're right there. What do they do, guys? A portion of the membrane, it buds out, okay? So this portion right there is what? Taken out and what? Turns into little vesicle, okay? It comes out, okay? Like they take a portion of the membrane. You understand what I'm saying? And then what? This guy goes in there. You understand, guys, here? So now, what happens, guys? Yeah, dude, it takes a portion. Okay, now, the membrane that was part of the ER now is what? Part of what? The vesicle. You understand what I'm saying? Okay? And now, so this one, guys, this is AP. This AP, they teach this too. Okay? Like AP, they teach that vesicle part too. And the membrane, that's AP, okay? It's just good enough, guys. Jericho, I don't think so. I mean, who knows? I mean, I don't know. No, guys. No, guys. Okay, they will probably teach you guys, I guess. But now, try to listen very carefully, okay? So how do they know where to go? Like, okay, so they transfer between the two endomembrane system by some what? Vesicles. okay? But now, how do they know where to go? How do they know where's what, guys, okay? How do they know where's what, guys? They put something they call V snare. This is not something that your teacher will teach you about, okay? They put a little protein marker called V, vesicle snare, okay? So V snare, they put these snares on them in the vesicle, okay? And then, so basically what it does is what, guys? It finds the target, okay? Which is what they call what? Terminus snare. They have something they call, guys, a T snare. So this is the Golgi apparatus, okay? And they got what, guys? A T snare. Okay. So what it does, so V snare and T snare, okay? The vesicles will go there and binds to it and what? Fuse the membrane onto what? Golgi apparatus. Okay? So inside will go into the apparatus. So this snare is what? Is there to give them the destination where to go. Okay, so now. So then what happens, guys? So yeah, this guy will go and what? Fuse into the membrane. Okay? Because like think about it, guys. That's the understanding of what, guys? Endomembrane system. They got the same membrane, okay? They can just fuse together and what? Put stuff in there. So keep in mind, so what they're doing is what? The membrane from the ER is what fused into what? Golgi apparatus. You know what I'm saying, guys? That's what endomembrane system is. Then what happens, guys? This guy now went in. Okay? So I tell you guys, your teacher's not gonna teach this, by the way, it's between you and me, okay? So with the Golgi apparatus, okay, now like you guys understand the vesicles, you know, going from the ER to the Golgi apparatus, guys. So the Golgi apparatus, guys, these are called the cisternae, okay? So you know, little sacs of the Golgi apparatus, by the way. So one thing good to know, guys, okay? This side of the apparatus, Golgi apparatus, guys, called the cis side, okay, cis. And that side is called the trans side, okay, trans and cis side of the Golgi apparatus, okay? So keep in mind, guys, this is facing the ER, trans is facing the membrane, okay? Why? I don't know why I don't know. So that's what it is, man. So now, and then basically the role of the Golgi apparatus is what, guys? So keep in mind, guys, a lot of these proteins that's being outbound for the secretory purposes, going outside the cell, is what a lot of times those are what, guys? Glycoproteins. Okay. What's a glycoprotein, guys? What are they? So they go a protein that's got a sugar on them, okay? Like carbohydrate on them, right? So typically what they do, okay, Golgi apparatus, guys, they don't mess up the protein, okay? Protein guys, they don't mess it up. They typically what, guys? So typically what? Modify the carbohydrate portion of it, okay? So there's a carbohydrate portion, guys, like, yeah, take one out, put one in there, guys. So modification a lot of times of the glyco portion of the protein, guys, you know. So that's what the modification they do, okay, in terms of Golgi apparatus. So they package it, like, for example, guys, like, think of the ER as a car factory. They produce the car, okay? And the Golgi apparatus is what, guys? Like that car dealership, okay? They nicely wash them, like, you know, put on better, like, you know, I don't know, aftermarket some crap on them, guys. Sometimes, like, I don't know, if you guys go see some of the like dealerships, guys, like they modify the engine too, guys. Like they tune the engine, okay? I don't know if you guys like Porsche or stuff like that. There's an RUF, a rough, you know, the engine tuners. They make the engines a little bigger, like, you know, make the bores larger, stuff like that. Okay, that's what they do, okay? So basically, that's what they do, okay? They modify, you know, some of the portion of the protein that's gonna go out, okay? And then what? Yeah, dude, same thing, right? Golgi apparatus, basically what they do, guys. Yeah, they produce the same way, the vesicles, okay? Vesicles, same way. V snare, T snare, guys, by the way. And what? Ship it out to what? The membrane, okay? And then what? Exocytosis, get the hell out, okay? And that could be what? Anything. It could be what? Insulin or serine molecule, whatever. So that's what they call, guys, an endomembrane system in a very high level. Okay, so these are what, so basically, what does it mean, guys? So you can kind of understand, guys, what's endomembrane system, guys? Their membrane is all the same thing, okay? So the phospholipid bilayer can be what, guys? Dude, it's all found here, okay? So yeah. So then, like, one layer fusing into the other guy, okay, as a vesicle. So there's a whole sector of the membrane, guys. The membrane, guys, we talk about it, guys, and then after we talk about all these like organelles and stuff like that. Okay? So what about guys? A smooth ER? What's their job, guys? Smooth ER. So a lot of these guys, these are secretary portion of it, guys, which is like produce a protein, get the hell out. Like this guy. Smooth ER, what's their role, guys? They produce what? Produce lipid. Lipid production, like steroid, for example, like cholesterol, for example, okay? They produce lipids. Like steroid, like hormones, for example, like, you know, like testosterone, like estrogen, for example, guys, and they're produced in what? Smooth ER. And also, guys, they also, you probably heard of guys, detox, right? Detoxification. By the way, okay? So they're like, if someone is a heavy drinker, by the way, okay? Then what happens, guys? In their liver, they have a very well developed cells with smooth ER. Because why? They detox like alcohols and the poisons and stuff like that in the small intestine and the smooth ER, by the way. Okay. And then they also are known to be a reservoir for calcium 2+, by the way. Okay. Calcium 2+ reservoir. They hold on to the calcium 2+ ion, by the way. Okay. That's one of the few things you have to know about smooth ER. I mean, very limited stuff, guys. Man, I don't know. You probably know I have to mention some of these guys on DAD, a little bit here and there. So out of all those, like, what's the AP guys? Like the 30S and like I'm sorry, the snare thing. Okay, that's one of the things, guys, that's a little bit higher on the AP side. Okay, other stuff, man, it's like same thing. I think. Yeah, and we'll see. Okay. And then the other stuff, guys, other organelles. I'm kind of rushing a little bit because I want to really finish this. Other stuff you guys have to know, guys. The lysosome. So lysosome is when you guys have to know a little bit is, lysosome has a membrane also as well, guys, okay? And then within lysosome, they always say this hydrolytic enzymes. They have a hydrolytic enzymes in there. Okay, so which means like enzyme that breaks down, okay? So the pH is around 5 inside this shell, okay? And it's surrounded by, you know, these hydrolytic enzymes. Whatever the enzyme that is, okay, it's very vague. It's a high level, by the way. Could be anything, okay. So what do they do, guys? What does lysosome do? So lysosome can be a lot of things, okay. So a lot of times, guys, if a cell, okay, brings in like a nutrient, okay, that needs to be broken down. So for example, guys, you know, like they have some nutrients outside the cell was brought in, okay. So now it's formed into a vesicle, as we know. And this thing is in there. So what happens, what, guys? The hydrolytic enzyme, which is lysosome, will fuse with the vesicle. Okay, so whatever the enzyme that they got in there is what, guys? It's gonna fuse with this guy and start digesting it. So which means like these two will start to fuse together. Because why? That's the endomembrane system. The membranes can just fuse together, right? And what? The enzyme will start to what? Attack and digest whatever that was came in. Okay, so whatever that is, it's gonna start digesting it. So typically speaking, guys, like a lot of times, like for example, like immune system, right? Which they have a macrophage. So they engulf it and digest it. Okay, those are all in part of what, guys? Lysosome. Okay, and also they what? They internally, like, you know, like digest stuff that's in the cell. For example, guys, in the liver cell, they got what, guys? Glycogens, right? So glycogens, I mean, with the proper signaling, what happens, guys? They break this down into glucose. Okay, they gotta have an enzyme that turns into glucose, and glucose can go out the cell into the bloodstream, right? So there's what? There's an enzyme also, a lysosomic enzyme, lysosome enzyme, that will perform this function as well. And I'm sure you probably will see the question, guys, as homework, okay? There's a disease, okay, where it's failed to, if the lysosome failed to function, okay, there are some conditions, you know, as a person. For example, guys, like Tay-Sachs disease is one of them, right? They failed to break down some of the lipids, for example, guys, and it affects someone's brain, okay? Because lysosomes failed to function. And also, there's you probably see this on your homework, guys. I don't know how to say it, Pompe, okay, disease or condition. This is where, guys, they failed to break down glycogen. Okay, so your cells, your liver cells failed to break the glycogen into glucose. Okay, because why? Lysosome failed to function. Okay, so imagine, guys, your body cannot break down you know, glycogen into glucose, guys, what will happen, guys? Yeah, you can't get much of energy, so this is a condition, right? So they failed, if someone has this, guys, by the way, this condition, they failed to live more than 2 years. Because the baby, if one of the babies got this, guys, they can't live more than 2 years, okay? This is all, guys, genetic condition, by the way, okay? Seems like, yeah, in DNA, they probably what, don't have the proper enzyme, okay, that does this function. So that's one of the things you have to know about the lysosome, guys, by the way, okay? Questions? Any ideas? It's a reading, guys. Read the textbook, guys, please. Okay, they got a lot of these stuff in there. All right, so the other organelles, guys, that we talk about actually is peroxisome. So peroxisome, okay. So I think their job also, guys. So you gotta read the textbook, guys. But their job is, guys, for the detoxification as well. So there are many jobs, detoxification. So and also, guys, like converting like a fatty acid into glucose also as well. That's what their job is, guys. I mean, oxidation as well. So usually what they're known for, okay? Like I'm sure you like, oh, peroxy, that's what? Hydrogen peroxide, H2O2, okay? They have some linkage with the hydrogen peroxide, okay? H2O2, because, yes, they do, because what their function is, okay? If you guys have, if there's like hydrocarbon molecule like this, okay? What they do is, as their catalytic reaction, by the way, peroxisome is not part of the endomembrane system. Okay, their membrane doesn't fuse into the other guys, okay, by the way. So they remove the hydrogen from the catalytic process as a part of detoxification, maybe modification of the fatty acid, whatever. Okay, they take hydrogen out, and they fuse this with the O2 gases, and makes it into what, guys? H2O2, which is hydrogen peroxide. So that's their function, by the way. Okay? So that, whatever this part of the product is, as a part of detoxification, or as a part of, guys, their major function that they're doing, guys. So they take the hydrogen out, guys, and feeds it O2 and turns into what? Hydrogen peroxide. That's why they call the name, guys, peroxy. Okay. And now, one thing to note, okay, this is some of the stuff that you guys have to know for the Olympiad. Okay. There's an enzyme in the peroxisome. There's an enzyme called catalase. Okay, catalase. Catalase, what do they do is what so H2O2, man, this is not very good for the cell. Okay, this is like toxic substance for the cells. Okay, you can kill them. So what they do is like there's an enzyme called catalase in peroxisome, which you break this down into what, guys? Back into H2O and O2 gases. Okay, so that's a little bit better for them. So that's an enzyme called catalase for the peroxisome. Okay, that's one thing to note for the peroxisome, guys, for you guys. Okay. And now in plant cells, okay, so it's something that you guys have to know for the Olympiad is that there is a organelle called glyco, okay, glycoxisome, okay. I mean, I might be wrong, guys, by the way. This is a reading like glycoxisome, something like that, okay. So, so what it does, glycoxisome, guys, is that they're a class of the peroxisome, but this is found in plants. Okay, these are found in plants and fungi, maybe, guys. Okay, and their job is turning fatty acids, okay, lipids, fatty acid, right, into glucose. Okay, that's their job, by the way. Okay? That's one of the jobs that glycoxysomes will have. Just so you know, okay. So this isn't the reading, okay? So nothing fancy, except I didn't see the term catalase in the reading. I don't know why. I thought they did, but I couldn't find it, by the way. I even went back to my old textbook to find this enzyme, but it wasn't there. But yeah, that's, I don't know. I found it somewhere. I saw it somewhere. I forgot where, but in the textbook guide, by the way. But you gotta know the catalase, because this was on the homework for the next one, by the way. Okay? Any questions, guys? Let me know. This chapter is very like straightforward. Okay, nothing fancy, except for like a couple things here and there. Because this is something you should already know. A lot of parts of it, by the way. And they talk about, guys, chloroplast and mitochondria. I'll push that for next week because, I mean, again, there's a whole chapter about the respirations and photosynthesis next week. Okay? And I'll talk about some more basic stuff, guys, in Sunday and Saturday class for the AP folks. Any questions? Let me know, guys. Questions, concerns? Let me know. I'm sure I'm not missing anything. Yeah. All right. So. All right. So. The next part of this, guys, just gotta jump something straight into, guys. So the organelles, guys, I'm gonna skip the chloroplast mitochondria and jump straight into cytoskeletons. I'm sure you heard some of these guys, by the way. Okay. Cytoskeletons, guys, they got microtubules, okay, microfilaments, and intermediate filaments, okay. So first thing first, microtubules. So microtubule, guys, is a cytoskeleton. So first thing first, guys, cytoskeleton. Basically, guys, I mean, how they found this out is that, like, we talk about all these organelles and like peroxisomes and lysosome and all that stuff, guys. So in the beginning, right? Nucleus and everything, like organelles and all that stuff, guys. Okay. So they think, okay, all these just float around in the cytosol. Okay. They float around. Okay. As like the boat floats around, you know, in water. So they later found out that was not the case. Okay. That's not the case how it works. Okay. So there are cytoskeleton, okay, in the cell, just like how we have a skeleton in our body, guys. We have an endoskeleton, just like how a skeleton our bones, by the way. So they also have what? Something called cytoskeleton, made up of what? Microtubules, microfilaments, okay, and also what? Intermediate filaments, okay, within the cells. So the first thing first, guys, microtubules. This is made up of what we call, guys, tubulins. Tubulin is the protein that makes up microtubules, okay? Textbook, guys, okay? So microtubules, guys, I mean, so basically the shape of the cell is what? It's all what? It's given by the cytoskeleton, okay? So it's like how your body has a shape, guys, just so you know, because what you guys have a skeleton, same thing, okay? They got what? These microtubules, filaments, and intermediate filaments, guys. They're the ones that what creates the shape of the cell of its own. And also, okay, what do these what, guys? The tubulin, which is like made up of tubulin, which makes up microtubules, okay? What they do is what? It provides, okay, like a rail within the cell for the organelles to move, okay? So what I'm saying is, guys, okay, if an organelle wants to go from here to here, okay? So basically what happens, what, guys? These organelles, whatever this organelle is, okay, they got a motor protein on them. Motor protein is a protein that needed to what? Be able to move. At the expense of ATP, they what? Collides on the microtubule in the cell. Okay, so at the expense of ATP, so you guys understand, guys, ATP, the expense of ATP, the motor protein makes it move. It's not like randomly just what, like, oh yeah, like floating around. No, it's these microtubules provide a pathway for the organelles to move at the expense of ATP with the motor proteins. So these motor proteins are on these organelles, okay, and attach themselves onto the microtubules and what? You have to move from point A to point B, okay? So these microtubules make them what? Mobility of what? Of the organelles, okay, with these motor proteins. And also, guys, microtubules also, I'm sure you guys, some of the stuff we'll talk about, guys, in the cell division as well, guys. In cell division, like for example, mitotic cell division, okay? And they're known to play a role, okay, of attaching microtubules onto the chromosomes during the cell division. Like you probably have heard, guys, like anaphase and metaphase and all that stuff. There's something, you know, like you know, hooking onto the chromosome and separating them, guys. Those are what, guys? Microtubules, okay? So within a cell, okay, so where do they produce microtubules, right? So for the animal cells, guys, there's a region called centrosome. Centrosome, like this one, guys, you're probably, I'm not sure, guys, how much your teacher will be talking about it, guys. Centrosome is a region within the cell, okay? It's a location on the cell, okay? Like near the nucleus, by the way, okay? And within the centrosome for the animal cells, by the way, okay, they got centrioles. The centriole. Centriole, so like I don't know if you ever heard, guys, seen it, guys, like they look like this, okay? Within the centrosome, the region within the cell, guys, these are called centrioles, okay? And they think that centrosome, like the microtubule formation occurs at what? At the centrosome portion of the cell, okay? That's where they generate these microtubules. And you gotta know specifically the structure of the centrioles, okay? Centrioles, guys, so they got, guys, so these tubulins, right, like microtubules, guys, this is a hollow tube, okay? So the shape, guys, like if I have a cross-sectional view of the microtubules, guys, I mean, it's like a little rod, okay? And now, if I take a cross-sectional view of it, guys, like this, it's what? It's a hollow tube, by the way. It's like empty, okay? And now, centrioles, okay, they got, so they say 9 plus 0, like you know, centriole, for example, they call 9 plus 0, by the way. And hear me out with this, okay? This is what it looks like. Draw it here. So, like, they got 9 of these guys, but I can't draw them very well, guys. So, they got 3 of the tubulins, okay, microtubules, okay, like in a row, okay, and then they got 9 of these guys lined up like this, okay, as a microtubule. So they're like centrioles, guys, okay, they got 9 of these lined up, triplet of the microtubules, okay, lined up, chained together. That's what they call, guys, centrioles, okay? So like if you look at these guys, they draw a box, but it's not really a box, okay? So where they got 3 of these guys, okay, these are microtubules, chained up, and they're what? 9 of these guys making a little circle. Okay, that's what they call centriole. And they got 2 of these guys vertical to each other. Keep in mind, centriole only applies for animal cells only. So centrosome, this is present for the animal cells and plant cells as well, okay? Centriole, where they got these configuration of the microtubules, they only have in the animal cells, okay? Your cell, my cell, but not the plant cells, okay? And they think, okay, this is where they produce microtubules, okay? That's where the location where the microtubules are being produced. Okay? And like, dude, like you guys all know, like centrosome or centrioles, okay, they play a role in what? Also cell division as well, okay? And also, guys, so the motility portion of it has also as well, right? For a single unicellular cells, okay, the cells that, like, you know, we have what, like your body has like millions of cells, right? But there are some organisms that has what, only 1 cell only, okay, unicellular organisms, right? Let's just like, yeah, what do they got, guys? Like, what do they have, guys? Like you know, some of the bacteria that you guys know, paramecium, you guys heard that, right? So, you know, and then Euglenias, okay, so you know, those are all what, like unicellular organisms, right? So, what do they have, guys? They got what, guys? They got cilia, right? And what? Flagellum. So they got these what, like, okay, flagellum or what? Cilias, right? And these guys are what? Made up of microtubules, okay? So, like, dude, like, if you look at it, guys, like, this is basically, guys, a flagellum, right? So if you take a cross-sectional view of this, okay, this is important, by the way, because this is something that they'll ask you. AP, they used to, but not anymore. Olympiad, yes, okay? So, a motile, moving cilia, this is important, guys. So, yeah, listen carefully, okay? the cilia that we're talking here, okay? These are eukaryotic cilia, okay? Keep that in mind, okay? Flagella also, like these are eukaryotic cells that we're talking here. If I take a cross-sectional view of this, okay, they look like this. Listen carefully, okay? These are known to what they call 9+2 configuration. 9+2, okay? For those motile cilia, it's important to understand moving cilia, okay? Like cilia that's known to what? Like that's created to what? Make them move, okay? There are some non-motile cilia. They call those primary cilia, okay? I don't know if it's in the reading, by the way. Their structure is a little different, okay? And keep in mind, guys, your prokaryotic cilia is also different in structure. They're completely different in structure, okay? So if you look at this, guys, 9-2 configuration means like this, okay? So they got these microtubules, okay? 9 of these are lined up like this on the outer portion. 1, 2, 3, 4, 5, 6, 7, 8. I'm gonna just run out, okay? 9 of these guys, okay? They're all paired up, microtubules, guys, by the way. And they got 2 in the middle, okay? That's what they call, guys, 9 on the outer portion, okay? And 2 in the middle, okay? That's what they call, guys, a 9-2 configuration, okay, of the microtubules for flagellum and also cilium or cilia, whatever you call this, okay? Usually in a cell, they have one flagellum, and they have multiple cilia, okay? So plural, singular, whatever, okay? You guys know English better than I do. So that's the typical structure, okay? But now, what they call primary cilia, okay, like paramecium cilia, right? They have what, guys? 9+0. They don't have the inner portion of these microtubules, by the way. Okay? They're missing inside a portion of it, okay? They don't have those things in there. So 9+0 is primary cilia. Make sure to understand, guys, okay? The prokaryotic cilia, they're completely different in structure, okay? They just have a very, like they don't have the microtubules there. And also, guys, keep in mind also, like they call this part, guys, wherever this cilia or flagellum is embedded into the body of the cell, that's called the basal body, basal portion of the basal body, the basal portion, like the bottom portion where it's connected to. So the basal body has a very similar structure to centriole, where they got what, guys? Like they got a triplet of the microtubules, by the way, the 9 +0, but it has triplet of those microtubules, guys, like centriole portion, centriole lifting microtubules, okay? So that's what can be found in the basal body. The basal body, they don't have the same 9-2 configuration, guys, but they have what? They have a very similar portion of the basal body as a centriole, like, you know, distribution of the microtubules, by the way. This is, I gotta, like, dude, I tell you, this is Olympiad. You gotta know this, guy, all of it. Like, primary, like, okay, 9-2, you know, centriole, you gotta know them, okay, basal body and all that stuff, okay? And also, guys, so this is in the textbook, okay, which I'm sure you guys want to read it. Between these two, okay, between these microtubule rods, okay, like, you know, this is one, that's the other, guys. Between these two, there's a protein called, okay, dyneins. And this is the protein, this is the reading, okay? So this is the protein that's what? Making them like possible to be able to what? To be able to have that motile motion, okay? So it's hard to explain, guys, with my limited drawing, guys, by the way. So guys, just to let you know, like this is one rod, like this is a rod of one of those, you know, like one of those 9. And this is the other guy, okay? And they're connected by this protein called dyneins, okay? They're connected by these proteins, okay? Think about, they gotta move, okay? So think about, these are what? These are protein called tubulins, like they're like rotating cylinders, okay? So at flagellum, what happens, guys? They gotta move, right? They gotta make that propulsion to move, right? So, but if you look at it, how are they gonna do this, guys? I mean, how are they gonna like curve? So this thing has to curve, like, you know, they gotta bend. How they're gonna bend is a question you have to ask yourself, okay? So, like what they do is what, guys? at the moment, at the expense of ATP, okay, expense of ATP, guys, so these are the protein called dyneins, right? At the expense of ATP, what happens, guys? At one point, one portion detaches itself onto from one of the microtubules, okay? They detach itself onto, and then what? And they make them start to bend, okay? And then once they're bent, like now they're like, you know, like, oh yeah, we're gonna bend, okay? And what happens, guys? And then at the expense of ATP, the phosphate group comes off, and what? And then they reattach themselves on there. So that's how they try. I don't know if you guys have to go that into much detail, guys. The book explains it, guys, with a better drawing. So you might want to go back and see what it is. But this is one of the protein that's at the expense of ATP, responsible for what? The flagellum and cilia to be motile, okay? And able to bend. Good to know, okay? So microtubules, guys, and you guys have to really, really know what they do. Okay? You gotta have a good understanding of, guys, what they're made up of, what their role is, especially these guys, okay? And then, guys, it's just not good. So probably, guys, like when you guys come for the AP Bio class, guys, I'll probably rush down and to make sure that we discuss the transports and the other portion because the time is running out. All right, so you gotta understand, guys. There's a reason I'm rushing, okay? Because we're in chapter 7 right now. You gotta go to chapter 50. So now, and I gotta do this, guys, before like what, October or something? You know, so it's why I'm like rushing, okay, like with these things, okay. But you gotta do your reading, guys, by the way, and then solve questions. You know, like I don't know. Okay, so now, guys, there's microfilaments. So microfilaments, guys. So microtubules are created by the protein called tubulins, right? But microfilaments are created by the protein called actins. Okay. So actin is something, I don't know, maybe you guys might have heard this before, I'm not sure, guys. I mean, it's a muscle, okay? It's made up of actin proteins, okay? Actin and myosin, by the way. Okay, sliding mechanism, by the way. So that's one of the things where you hear actins quite a few times, by the way, okay? Microfilaments also, guys, it maintains the structure of the cells, okay? This is part of the cytoskeleton. But actin is a lot smaller in size compared to tubulins, by the way, okay? The actin microfilaments are a lot thinner compared to the microtubules. So, you know, they're throughout the cells, of course, guys, so microtubules. And their role of microfilaments, guys, by the way. So we'll talk about it a little bit, guys, by the way, but during the cell division, okay, in the cell cycle perspective, okay, there's a cell cycle, guys, I'm sure you guys have seen this somewhere, guys. There's a G1, there's S phase, G2 phase, by the way, there's M phase, there's a C phase, okay? So that is where, guys, they divide the cytoplasm during the cell division, okay? So when the cell divides, okay, they have to divide the cytoplasm for the animal cell division, okay? And now there's a portion of the cell division where they call, guys, a cleavage furrow. They create a cleavage furrow where that's the sort of a last step of the cell division, okay? Where, you know, microfilaments, that's where they play a role there, okay? They squeeze out between the two cells, okay? And they're trying to make a gap between the cells, okay? That's called a cleavage furrow. So when the cells are about to divide, you know, like they look like this, guys, by the way, and they create this cleavage furrow on both sides. It's like a zip tie, okay? Like zipping it out. And that's probably done, guys, by the actin of the microfilaments. That's the role. And also, guys, microfilaments, also this actin part of it, guys, they play a role also in one of the junction between the cells. So if there's two cells, so, you know, epithelial cells, okay, the cells like, you know, in your lining in the stomach, for example, guys, on your skin cells, by the way, two cells have a lot of junction between them, okay? So one of the junctions that involves the actin is adherin junction. So it's one of the junctions. So there are 4 junctions to know, guys. There, adherent junction. So this is N. There is a desmosome. There's a tight junction. It's all in the reading, guys. Okay, except for this guy. Tight junction. There's a gap junction. 알아야 돼. gotta know all of them. Okay, the gap junction, tight junction, desmosome, and adherent junction. Okay, you gotta know all of it for the Olympiad. Also for the AP. AP, you know, the first 3 maybe. So adherent junction. So adherent junction. Okay, they use actin. Okay, for the anchorage of the 2 cells. Like, dude, 2 cells must anchor together. Okay, dude, like 2 cells, like, dude, like they can't really move around, right? So they anchor the 2 cells together. That's one of the junction called the adherent junction. Okay, so they use what? They use actin to stabilize. Okay. So that's what these are one of the things they utilize. And examples they use, guys, you know, the microfilaments for within the cells. Okay. And now the other one, which is the we call, guys, intermediate. We don't really talk about this one too much. Intermediate filaments. So this is there's no particular like protein that produces it. But the example, guys, is like, for example, keratin. So keratin is one of the protein example, guys, that can create intermediate filaments. Okay. So same thing, guys. The structure of the cells, you know, is maintained by the intermediate filaments as well, just like microtubules, microfilaments, them as well. But intermediate filaments, guys, their role is to stabilize and anchor down the organelles. Okay, it's like nucleus. You know, they cannot really move, like, you know, from that, you know, middle portion of the cell. So these intermediate filaments kind of anchor these, like, you know, organelles and nucleus down to the position of themselves. Okay. So now, well, intermediate filaments also, guys. I don't know if you're interested, guys, by the way. Inside the nucleus, okay? Inside the nucleus, okay? The bottom, like, inner portion of the nucleus, guys, they got something they call nuclear lamina, okay? And that is also what? Created by intermediate filaments, okay? By the way. And also, if I'm not mistaken, guys, by the way, okay? The desmosome, which also anchors the 2 cells together, desmosome, guys. It's like a welding of the 2 cells together, make sure, you know, so that they don't really come apart. Very similar to the adherent junction, by the way. They use what? They use actually intermediate filaments. Yeah, do you guys think, okay, so would that be ever my test? Absolutely yes, just so you know. This test, guys, is crazy, okay, so you know. I tell you, this exam, every little detail is important to this test. That's why I'm saying this, a 2-hour class, guys, like, you know, I just give you like a bunch of information, guys. It might not be enough. Okay, you gotta do your freaking reading, guys, okay? And even like the reading I'm giving you guys, that might not be enough. That's why I'm giving you more information, guys, because so you know. So anything can be the test, guys. Anything. And if you don't know it, guys, you can't freaking solve it because it's not something you can just figure out on the fly. So these are what we consider, guys, as you know, like microtubules, you know, microfilaments, intermediate filaments, guys. These are all part of what they call cytoskeleton, okay, which creates the shape of the cells, which makes the shape of the cells, and you know, all their functions gets involved as well, okay. And now, in terms of, guys, a few other things here. So these guys, you have to know, guys, what these are. Desmosome, we talked about it a little bit, guys, and the tight junction, gap junction, and adherent junction. You gotta know what they are. Okay, so we talked about, guys, desmosome. It's in the reading also, guys. I mean, this is like anchored the 2 cells together, guys. Okay. AP, they asked this as well. Okay. This one is not really for the AP, by the way. Okay. Tight junctions. Okay. So tight junction, guys, is what the name kind of states. I know some stuff about this actually. More detail. Okay. They call this, guys, also occluding junction as well. Okay, occluding junction. Tight junction, I highly doubt, guys, okay, that's what they're gonna say, occluding junction. Because I mean, again, so basically, guys, tight junction is where the 2 cells, right? You got 2 cells here. So a lot of times, guys, you're like, for example, you guys eat water, okay? Water should follow esophagus, your stomach, your intestine, and goes back out, right? So, so which means the water that you're drinking, the food that you're eating is what? They cannot come out of your, the canal, okay? They cannot come out into a peritoneal cavity. If anything leaks out, what happens, guys, is there'll be a major infection, okay? So to do that, what happens, guys? Your canal, like stomach and small intestine, large intestine, whatever, they must be what, guys? They must be watertight. How do they do that, guys? They got the cells have what? Tight junctions, okay? Like it's like what? They staple these two cells together so well it doesn't what? Leak anything. It doesn't really leak water. Sometimes called this what, guys? Occluding junction. Okay. Bunch of proteins, guys, okay? Occludin, that's the protein, okay? That's what occluding junction, okay? And then a gap junction, guys, okay? Is where there's a cell, okay? Gap junction, guys, okay, is where, you gotta know this, guys, okay? So it kind of makes so that two cells can communicate with each other and transfer the material between the two, okay? So for example, guys, like hormone and like sodium ions or whatever that has to go across the cell that's nearby, they can go through a channel called gap junction. Okay, and gap junction is created by a protein called connexin. Okay, I mean 2N. Connexin. Okay, that's a little channel they create between the two cells, by the way. Okay, so yeah, so that's what a few things you have to know, guys. And you gotta know. There's definitely a question of the homework, guys, that relies on one of these terms that I just talked about. You want to take it, so this is not in the reading, guys, by the way. Okay, zero junction, guys. It's not the reading, so make sure to say that, okay? Questions, So I would like to, guys, have the AP class go a little ahead of you guys, by the way, like, but the kids are there, I don't know. Yeah, so we'll see, guys. So that class must come first before this, okay, so like, we should have talked about some of these guys, and then, like, we go a little on top of that. That's the preferred direction, guys, because this is like, we didn't talk about this yet, okay, in that class. So at the same time, but I can't like lag back because like we gotta go. Okay, we gotta push it. So we'll see. I try to time it right so that the other class should go a little quicker than you guys, okay? So that you guys, like, when I talk about these things, you have a little understanding what else is going on, okay? Or I can give you the reading. I mean, you guys can read a little further ahead if you want to, if you have time for it. I mean, I don't know, but I don't know how much time you guys have over the break, guys, by the way. But any questions, let me know, guys, so far? Questions, let me know. I mean, the one thing that's remaining, guys, that we have to discuss in this reading is ECM. I mean, you can well, like, you can do the reading, guys, okay? It's called the extracellular. Okay, matrix. That's what it's called. ECM, okay, extracellular matrix. So, so what this is, guys, ECM. So compared to the guys, the plant cells. So what's the major difference between plant cells and animal cells, guys? Plant cells have what, guys? They have a chloroplast, and what? They also have what? Cell wall. They have a cell wall, right? So now, the cell wall, guys, for the plants. So they have a primary cell wall. Okay, cell wall again is made up of cellulose, okay? And then they have what, guys? They also have the primary cell wall is very thin, okay? This is outermost, okay? They're very thin. And they also have what? They also have secondary cell wall. Okay. So secondary cell wall, guys, can be much thicker, okay? So it's much stronger, and they have a protein called lignin, where they strengthen, okay, the secondary cell wall a lot, okay? And then below that, they have a cell membrane, okay, plasma membrane. This is the plasma. So primary, secondary, plasma membrane. Okay, that's how they hold, okay? And also they have a very large vacuole. They have a very large, like, central vacuole for the cells, for the plant cells, by the way. So that's, like, structurally they're very different, by the way. And now these cell walls, what happens, guys? They provide what? Protection for the cells, right? So like, yeah, they provide a little casing around the cell that what protects them. Okay. So then, what protection is what? What protection is what? They protect them from what? Lysine. If water goes into a cell, what happens, guys? Right? Cells can burst. They're done. Okay. But now, because what? Because cell walls, what happens, guys? They give them what? Rigidity. Okay. That's called turgid. Okay. Rigidity, right? So, but what about the animal cells? Do they have anything like that, guys? Do they have anything like that, guys? Like, can water go in and can they survive? You know, that's a question, right? So they have a different mechanism, guys, by the way. But again, what's around, what's above the cell membrane for the animal cells, right? That's what they call ECM. So for the animal cells, they have a phospholipid bilayer. Okay, right above it, they got something called ECM, which is the extracellular matrix. Okay, so this is nothing fancy, guys. Okay, so they've got a bunch of what? Collagen, protein. So it's just a bunch of collagens. Okay, this is a protein, by the way. Okay, this is as we know, guys, structural protein. Okay, kind of wraps around the entire cell. Okay, so these are the collagens. Okay, so they got, guys, a protein called integrins. Okay, protein on the membrane. Okay, and they got the connecting protein called fibronectin. It's in the all reading, guys. Okay, and now these are tied to these collagens. Okay, so like these are all over the place. So, and also, guys, they have a proteoglycan. Okay, which is like, so all of these guys, proteoglycan and collagens. I mean, collagen maybe not. Proteoglycan. These are like carbohydrates. Like these are, as you can see, guys, proteoglycan, a protein with the carbohydrates on them, guys. So they're like, it kind of wraps around, something like ECM, kind of wraps around the entire cell, okay? Sort of, I mean, so they don't have the cell wall, but they got the ECM there, okay? What's the purpose of these guys? Purpose is what, guys? Yeah, sometimes like they do a signaling, okay? Like, if, for example, guys, signal comes in, and then they kind of identify what kind of signal it is and such, guys, and sometimes provide what protection for the cells as well. Okay? So what the ECM is, I mean, it's good to know in terms of what it is. So you have to know the terms. Extracellular matrix. You got to know what it is. Okay. For now, in terms of discussing this. Okay. So they don't have a cell wall, but they got ECM, which, for now, is what it is. Good to know. But this is my concern. Okay. So you might be whatever, man. But it could be on your test. That's why, like, oh yeah, what's on ECM or something? Okay? Like, there could be a well as a question that you can look at. Any questions, maybe? And that's high level, guys. What's gonna be on your? This is chapter 6 in the reading. Okay, a lot of things we talked about right now. Chapter 6. Okay, I definitely rushed it 100%, guys. 100% rushed it, guys. And the chapter 7, I couldn't even start, guys. By the way, so a lot of information, guys. Okay, and try to squeeze everything into like one session right now. So a lot of information, guys. But like right now you can't even do the homework, guys. So like I would hate to, guys, just make you read it and just do the questions. So yes, for now, guys, like you gotta read the chapter, guys, please. 6 and 7, okay. And if you want to go over, like, don't mess it up. Like, don't like get 0, like half of the questions wrong. If you don't feel confident. Don't do the homework, guys. Okay, it's fine. Like did I assign the homework yet? Or I'm not sure. But the cell organelles and membrane. Okay, if you want to do it, guys, read the chapter, and if you want to do it, guys, 6 and 7, go ahead and do it. Okay. The cell membrane and transport. Yeah, I don't know if you require a lot of information on that. I'm not sure. Okay, if you want to go ahead and do it, go ahead. But probably next week, guys, we'll have to discuss the transport stuff. And then, I don't know, I'll try to go a little faster on the EP class so that we cover more on that one compared to this one. Okay? All right, guys.